So still in the SAM and BAM format, let's talk now about the SIGA string. So this is one of the most important fields, which contains a sequence of numbers and letters. They represent the information on the basis alignment. So each letter would indicate the description of the alignment. So for example, you have an M that would indicate that this is an alignment match. I as an insertion, D as a deletion, etc., etc. Then you have also R next and P next, which are the name and the position of the next or the mate pair. Then the T-len, which would be the observed length, then the sequence of the read itself, and the quality as it was appearing in the FASTQ file. So be careful because, in fact, in these kind of file formats, redundant sequences, so like duplicate reads, can be flagged, so marked, and removed from the files. So in fact, they represent kind of multiple reads that are aligning to the same unique DNA fragment. So these could be possibly amplified by PCR during the sequencing workflow, and this could induce a lot of errors. So it is critical to identify and remove these duplicate reads prior to variant calling. So this is an important step that is called marking duplicates at this stage. So we saw what the index was for the FASTA file. There is also a need for that kind of companion file for the BAM file, which is .bai. So you would have a .sam, so .sam, .sai, then a .bam.bai, and this would indicate that this is an indexed file. This is usually computed by SAM tools, which is used to view the BAM files, to retrieve data from them, to index them also. And when these kind of indexing files are really important for questions about one specific region. So if you have an index, you do not have to search for the whole file again, you just have to look at the index, and then you do not have to download the entire file, you just have to take the index and then have a look at the part that is interesting for you. So indexing aims to achieve a fast retrieval of alignments over a specific region. So the reference sequence have to be indexed too, as we saw it. So this is useful for visualization tools that can help you see these files directly, and a good example of this kind of software or tool is called IGV, that stands for Integrative Genomics Viewer, that is hosted by the Broad Institute. You have a link here to it. We won't have enough time, unfortunately, to cover it, but I just wanted to let you know that this is a software that uses the BAM format and that allows you not only to visualize reads on the genome, but also to combine it with the genome annotation, so basically the GFF file. So these tools allow you to zoom in and out the region in order to balance between a whole genomic view and a more in-depth analysis or single variants like SNPs. Other useful file formats exist and they can be used also. You have here the example of the BED format. This is a tab delimited text file that would define a feature track, and it is used to store information like genomic starts and ends for chromosomes, etc. I won't have enough time to cover it here also, but you have a link to it in order to have the details of this format if you want. Okay, so now that at this stage you have computed your alignment and that you controlled it and that you got rid or removed all the reads that are unwanted, then what you have to do is to evaluate the genomic coverage. So careful, because here you have three main concepts that are usually associated to coverage. They are the fold coverage, so this is basically a number that is followed by an X. maybe this is the one that you most hear about, because it's the most basic metric for genome completion or genome coverage. It is the number the mean number of times each base is seen. So, for example, if you have a 20x coverage, that means that on average each base has been read by 20 sequences. So this is the basic metric of it. Then you have also a breadth of coverage and the depth of coverage. Breadth represents more the percentage of the genome that have reads aligned to it, whereas the depth would more often correspond to the number of nucleotides that are contributing to a specific portion of the assembly of the genome. So this means that these terms often refer to, this is basically the same information but seen from different points of view, and you have the breadth that is being more calculated over the length of the read, whereas the depth would be more on the coverage that each base has. So once you have your read map to the genome and you would like to know where your genes are, where the genes are having a variant on them, so you take the genes that are annotated in the genome, and then the next step is to search for the common DNA mutation that would define the changes that you have in the genome. So, for example, if you look at the structural variations, then this would include different types of variation. We will be seeing them in the next slide. And by definition, because they include a huge number of variation, they can also include the CNVs, which stand for copy number variation. So we can talk about gene CNV or even chromosome CNV, but mainly this is used for genes. So gene CNV would be regions of the genome or genes that are affected by some kind of mutation. So basically you have a CNV when you have less or more material than what is expected in the reference sequence. So you also have detection of variant calling that includes mutations of only one nucleotide, and that would be the SNPs, so single nucleotide polymorphisms, or more than one nucleotide, and that would be the indels, so insertions and deletions. They could be detected thanks to the correct mapping of the reads, and this correct mapping allows you to screen for CNVs, for SNPs, for small indels, and the longer SVs, which are structural variations, are discovered by unexpected distances, in fact, between the two pairs of your paired end reads, so the second pair basically compared to the first one. And if you remember, we talked about this a bit as an advantage of choosing paired end sequencing over single end sequencing. So you have a general overview here of these events that constitute the common variation event that we look for using NGS. So they could affect large portion of the chromosomes, and you have here an example of the SVs that can also include here other kind of events. So SVs are basically all these except the single nucleotides, but these are very different definitions. You can find different definitions in the literature of that. So if we talk about gene CNVs, you have here an examples of them. If you consider the situation here, what is in the orientation of the ABC in the reference, you have here a duplication event, you have a translocation from elsewhere in the genome, you have also here a deletion of B, you have an inversion of B and C, etc. And the CNV so could count as part of these structural events. And then you have small indels that can be 2 base pair or more, or that can also touch just or affect just one base. And then you have the SNPs that are single nucleotide polymorphisms. So an information about SNPs and CNV would be stored in a VCF format. This is a text file format that contains 8 required fields that you can see here highlighted with that red box. So you have the chromosome position, the chromosome identification, then it's the position of the mutation, which is the POS, the second column, ID would list the unique identifiers, so if they are available. So of course, if you have, for example, a database related to SNPs, then you would have a DB-SNP variant, and you would usually find that RS number. You have also reference base, you have an alt column that would list the non-reference allele, etc. So what you would basically need to have a look at is this base that is the reference one, and the alt column that would contain the alternative or the mutated base. So you have here just a single mutation, you have here insertions or deletions. So Dr. Colin Saunders would be covering this in one of the next sessions. So, and then, of course, if you arrive at this stage of the analysis, then let's say that you are proud of having generated all this valuable data, but now you still need to biologically interpret your data. So this might be one of the, I would say, the funniest part, but it's quite complex because you have to compile all the data, you have to combine it in order to retrieve the important information out of it. So there are several web-based interfaces such as Galaxy that would ease the process of generation of variants, but it has problems in in the way it handles very large data sets, and they also lack flexibility for annotation. Other tools exist like VariantDB, which are web-based annotations. So these are available ones that will help you not only to annotate your variants, but also to correlate them to a certain level of biological information. So there are several of them. There are others that are packages that are available through the command line interface. The goal of all this is to come up with a potential role of your variants according to their functional impact, the pathogenicity, the severity, and you would use basically the gene ontology, which is based on the biological process or molecular function or cellular component that is associated to each of your gene, then identify pathway, then predict what these pathways will have as a role. Then I would also recommend you the use of some kind of tools or databases that are available. Once you come up with a specific set of genes that are of interest for you, and you have here the example of the DAVI database, which is database for annotation, visualization, and integrated discovery. And this allows you to switch between several databases and the identifiers of several databases, like, for example, if you want to go from Interpro to an ensemble ID, etc. And this is really, really useful. And it also allows you to connect your genes to biological processes, so the gene ontology annotation, etc. And there are plenty of them throughout the literature. So, as a conclusion, what we could say is, first, you need to be clearly focused on the question that you would like to address. So, the design of the experiment, the library construction, and the pipeline of analysis would heavily rely on this. Then you need to define the platform that you would use. So, each one has its own specificities that needs to be understood. You have different technologies, you have different evolving rates of these technologies, so make sure you keep updated about them. You have the possibility to combine different platforms if they give you different kind of informations. Then you need to know what are the input files and output files, and we saw that there is a great number of files that is needed at the various steps of the analysis, each having its own specificities. So some are, to render things even more complicated, some are 1-based counting, some are 0-based counting, etc. So we've seen all that. And you have to know that programs usually they just know how to cope with it, but just make sure that you understand all this. So I hope that with this I have brought some light on this quite complicated and rapidly evolving field that I try to simplify as much as possible today, and I hope this was valuable for you, and I thank you for your attention.